Hi, my name is Julie Thompson, and in this video podcast, we're going to be discussing bovine spongiform encephalopathy, or BSE. That might sound like a mouthful, but it's probably a disease you've heard of before. It's more commonly referred to as mad cow disease. It's an infectious disease, and when you think of an infectious disease, my guess is that you're thinking of viruses and bacteria. But in fact, BSE is caused by an infectious protein, which is known as a prion. How do cattle get mad cow disease? Well, if they're fed contaminated remains, so if you give them meat and bone meal, which contains the ground up remains of other cattle and sheep, if those animals had the prions will be transferred to the cattle that eat the meat and bone meal. It turns out that the prion is actually a misfolded form of a normal cellular protein. So here I've represented the harmless form of the protein with this box, and the same primary structure, the same amino acid sequence makes up this protein, the harmful form. It just takes on a different shape. It changes the folding of the protein. So the normal form of the protein is referred to as PRPC, and the misfolded form, the harmful form, is referred to as PRPSC. And PRPSC, this is the prion. Now, a key aspect of prion diseases involve the action of the prion as a chaperone. So when you think of a chaperone, you probably think of an adult that's chaperoning something like a high school dance or other birthday parties, something like that. And the chaperone is making sure that the children behave properly. Well, the cell has chaperones for young or nascent proteins. And so what you see here is a nascent polypeptide chain that's just been made. It hasn't yet folded. And what the chaperone does is the chaperone will provide a nice little environment to ensure that that protein, as it folds, that it takes on the proper shape, the proper conformation. So in general, when we think of chaperones in cells, they are proteins that promote the proper folding of other proteins. Now, sometimes you get a bad chaperone, a chaperone that's not really doing its job and that is influencing others in a negative way. So hopefully we don't have this at a dance, but we do have this in the case of prion disease. So more broad, we could take a more broad definition of a chaperone and call it a protein that influences the folding of another protein. Prions fit this definition of chaperones because they influence the folding of normal healthy proteins. So back to our PRPC, this is something that you find in normal healthy neuronal tissue. If PRPC encounters or bumps into PRPSC, what happens is that the PRPSC influences its folding, and it causes the PRPC to take on the harmful form of the protein too. So this can keep happening. The other thing about this protein is that the misfolded form, the PRPSC, it's hydrophobic, and it has these hydrophobic amino acids that don't like to be in the cytosolic environment, which is aqueous. Instead, it likes to congregate with other hydrophobic PRPSC proteins. And so not only do you get the formation of new prions, you also get their aggregation. And so that's what you see here. You see a mix of normal protein and prions. And the prions, if they happen to come into contact with the normal protein, they influence the folding in such a way that the normal protein takes on the prion conformation. And you also notice that the prions are aggregating and forming these fibers, or sometimes they're referred to as plaques. We know that these plaques aggregate on the outside or on the membrane of neurons. So this is neuron tissue from an animal that has been inoculated with a small amount of prion protein. And then the researchers wait some time, and then they go back and take the neuronal tissue and they stain it. and they're staining specifically for PRP-SC, and the stain in this case appears in brown. And what you can see is after inoculating the animal with just a small amount of prion proteins, they've accumulated quite a bit of this PRP-SC, and it's surrounding the neuron. So if we kind of look at this in a cartoon version, the normal cell, it has PRP-C that's on the membrane of the neuron. If you inoculate the animal, or if the animal becomes infected with a prion, the prion is going to turn those normal proteins or misfold them into the PRP-SC conformation, and you get this aggregation of prions around the neuron. Now, some of these PRP-SC proteins can also influence nearby neurons and start to change the proteins there into the PRP-SC form as well. These aggregates are not good for the neurons, so mechanistically we don't completely understand how this works, but we think that these prions that aggregate on the outside of the neuron, they give the cell the signal to die or undergo programmed cell death. And so these proteins that have prions surrounding them, they're going to die. And if you think about the overall architecture of the tissue, it's really going to change. So this is healthy, normal brain tissue compared with, on this side, we've got the brain tissue from an infected animal with BSE, and you can see that the death of the neurons leaves behind these holes, and the tissue looks sort of spongy, which is why this disease is called bovine spongiform encephalopathy. Okay, to summarize what we've learned, we know that BSE is a disease, it's an infectious disease, but it's caused by a prion. And in this case, the prion is called PRP-SC, and it's just a misfolded form of a normal cellular protein. And the PRP-SC is acting like a chaperone because it's directing the folding of the normal PRP-C, the normal protein, and it converts it into the harmful form. It converts it into a prion 2. And eventually, enough of this PRP-SC aggregates and forms fibers that are going to lead to the death of neurons in the tissue, and it's really just destroying the neuronal tissue of the animal. So as part of your task, you're asked to think about some recommendations that you could make to prevent the entry of BSE into the food supply, or prevent it from being introduced into cattle. And I think some important things to think about include, how do cows acquire BSE in the first place? So we said that if we feed cattle meat and bone meal, there's the potential for them to ingest the infected carcasses and therefore get the prion that way. I also encourage you to think about the resistance of prions to standard sterilization techniques. So even something like autoclaving is not going to be enough to destroy a prion, which tends to be highly stable. And so that's something to keep in mind if you have an infected animal that dies and you've got this carcass that contains these prion proteins. What could you do to dispose of this animal that's completely going to obliterate the prion proteins? All right, so I hope that video podcast was helpful. WGU students, feel free to contact us with questions about this or any of your other tasks at biochemistry@wgu.edu. You can also find us on Twitter at WGU Biochem. Thanks for watching.